Okay, thank you very much for everybody who is here after this long and very interesting day. Here is the agenda. It's going to be very hectic. You don't hear the mic? Is now better? Okay, thank you very much. Welcome, everybody. We are starting this hands-on session. There are going to be a lot of pieces into it, so just to make some order into what we are doing here. What we are going to do here in this hands-on session is to share with you two open-sourced models from IBM. We will share the Hugging Face, the GitHub, and how you can run them on the cloud. So we will have all of these pieces, and we will close with drug discovery workflows to show how it can be integrated within the system. If you just came and you want to sit, there are seats in the front, and you are more welcome to sit. So to make it very short, what we do today, and very optimistic, today we code, tomorrow we cure. That's the aim, that's the challenge. Let's start. What we have in terms of the technology that we are going to share with you is a biomedical foundation model set of technologies that we have created at IBM. Two models that were open sourced in this very week will be presented today. We are using these models in collaborations with different industry and academia. It aims in discovering targets, small molecules and biologics, and it aims in helping pharma and researchers in accelerating their science. We heard today a lot about this need, so I'm not going to elaborate at all. When we think about the user, we have three different persona, and you will see that this session that is very loaded, it is exactly because of that, because we want to provide input and help for the different personas. So we think about people who are machine learning researchers, engineers, who would like to play with the models in the typical way that machine learning experts are working with models. So you might want to download the weights from Hugging Face. You might want to fine tune with these models. You might want to have code that exemplify how to use the models. All of that will be presented today. You might need to scale up, so it might be insufficient for you to work on your laptop because you have large data sets for fine-tuning, or you want to retrain to have more data with the model that you care about. And for that, how to scale up, we will show through the OpenAD. And if you have in mind a commercial setting, you want to understand how these type of technologies can become part of a whole workflow within a system, then we will illustrate and demonstrate that at the end of this session. So I'll pick the very first model. As I said, we have two models, and we will show them from different angles. The very first model is based on a multi-aligned, okay. Phil told me not to do it, and I'll try again. Not to click twice, okay. So it aims at the very first model, we call it the architecture, the approach we call MAMMAL, which is a molecular aligned multimodal architecture language. And the model that we have generated from that is based on 258 parameters, and you will see the full name represents, the name of the model represents the different things that we captured within this model. It's a model that is similar in ways to BERT, meaning it's encoder only, but at the same time, it can also be encoder-decoder like T5. So it's some kind of a novel extension of T5 model that enables integrations of different sequences from different types of data. We capture data of small molecules represented by SMILES. We capture data of proteins represented by the amino acid sequence. And we capture data of gene expression, represented by the indicator that is the name of the gene and the expression level of the gene. And the model is extremely flexible in multiple ways. As said, you can benefit from it. You can use it through the encoder only. You can get the encoder results and generate from that. And you can also have encoder-decoder and get new sequences from the prompt sequence that you offer. And it can also integrate projections of other models into the input of the model. And the full details are available in a paper that was just launched, and you can find it in the GitHub, and it's in archive, if not now, then tonight. I mean, it's already submitted to archive. And the use cases are multiple. We tested that against many different tasks. Remember that we talked about learning from small molecules, proteins, and gene expression. The learning was done with the regular infilling, masking, all of these regular pre-training tasks, as well as unique tasks like protein-protein interaction pre-trained tasks. So learning from given two proteins, would they interact or not? And the testing can be done in all of these areas. So we created a test of drug-target interaction, where you want to predict the interaction. We created a test of infilling, where you get a protein, but it's missing parts of it, and you want to predict the missing amino acids from the sequence of the protein. You can see, and again, the full list of all of these tasks will appear, are appearing in the paper. You can see the different tasks. For each of these tasks, we identified what is the state of the art. You can see the references appear in the paper. What is the performance of the state of the art by whatever algorithm out there, because there's no one algorithm that provides the answer to the diverse set of questions that we tested. So for each of these specific downstream tasks, there's a different state of the art. You have the references. And we compared our performance, the model, the multi-aligned model performance, with the state of the art. So you have here two curves in the radar plot. One is blue, which is IBM Blue, what we generated. And the other one is the state of the art based on the different references. In each of the comparisons, the comparison is made on the exact same data split. And you can see that in many of the tasks, we outperformed state of the art. What I want to show you now is how to get access to this model and how to use the model. And I'm going to show you two different things. I'm going to ask Phil, so I won't do any mistake, to take me into the browser. So I'll need to take you in the browser from here. Hopefully I can do it well. I don't know that we started with the right one. I don't think so. We should start with the Hugging Face one here. OK. So this is the model available, the model weights available in Hugging Face. You can see that we have the exact same pictures with information of what the model performance is and information about how the architecture looks like. And there's no way I can read it from here, so I need to remember. You can see here example of how to import the model weights. You can see here how you import the tokenizer. There's specific token elements that has to do with the query. They are mapped into different elements that we can analyze interaction between proteins, for instance. So it's mapped into two different elements. And you have here the citation. You can see here, I don't know where my cursor is. So you can see here the two models, the fine-tuned models that you can download their weights as well. And I think beyond, and you can see here that it's 458 million parameters, this model, and the name is representative of the exact things that we downloaded, that were used for the training. So it's omics for gene expression, BL for biologics, SM for small molecule, multi-aligned T5-like encoder-decoder model with 458 million parameters. The intent is to release more models from this family, and actually there is a collection where you can see all of our models. You have a link here to the BioMed collection from IBM. Now I'm back to GitHub. So we have a BioMed SciAI GitHub where we share all of our code from the past few years. And in this week, we created this repository where you can see the contribution of these 7 researchers in the right here. Let me just see how I... And what we offer in the repository, it's a README with examples that show exactly how to load the weights and to fine-tune. So there is an example of fine-tuning of protein-protein interaction. This very first example, let me just get to the example. The protein solubility is not the first one. I think I just skipped the first one. The example of protein-protein interaction is unique in the sense that it has a task that is identical to the pre-trained task, and therefore, of course, the likelihood of success of such a fine-tuned model is much higher than many of the other things that would be new tasks. And you can see in the downstream tasks, there are other ways, other examples of how you can run downstream tasks, and you see the drug-target interaction. There's the protein solubility, and I think there's classification of cells. The flexibility of the model is much higher. There are unique tokenizer. Everything is documented. If you have any question, any issue, please do interact with the GitHub, provide the issues, make your comments. We would love to get feedback from the community. And with that, I'm done. Thank you. I'm inviting Partha. Thanks.
Yeah. So the second foundation model that we are introducing today is called multi-view, BioMed multi-view. So let's talk about it a little bit. So as we learned today that small molecule space is really big, 10 to the power of 64. Many of you might already know it. We believe that no single foundation model may be able to fully capture this really vast, unimaginable distribution of data. And there are 3 dominant approaches today. One is to take a sequence-based approach where you take a SMILES representation or some kind of a line encoding representation of a molecule, and then you divide it into smaller tokens, and then you learn this conditional probability for each token given the surrounding context. This works really well for NLP, and this has been attempted for learning representations of small molecules. There are many architectures. I can cite like 50 of them. Notably, there is an architecture called MoleFormer, there is Chemberta, there is BOT SMILES. You can go on and on. It's a dominant architecture. But that's not the only way to do it. Another popular way to do it is to take the 2D image representation of a molecule, and then you can learn from it. The advantage is that now you have a convolutional type of architecture, which can capture really unique things, because a picture is the picture. You get a global view of the molecule. You can learn things like symmetries and reflections and so on. So that's another way to do it. A third way to do it is to take a graph approach. A molecule can be visualized, thought of as atoms and bonds, even though it's a model of the molecule. There are different levels of theories, but this is one, right? And you can featureize it. And GNNs have been doing this for ages. And there are recently graph transformers also. And they are also powerful. So what we thought was, since the space that you are trying to capture is 10 to the power of 64, why not build all the 3? Because it's very difficult to compare foundation models because they are trained on different sets of data. So all these 3 foundation models that you have built are all trained on the same 200 million data points from PubChem and Zinc. And we have an architecture that combines the latent spaces in a way that is very task specific. We explored several different late fusion architectures for combining the latent spaces, and we are exposing all of them through our API, and we are recommending one for a particular usage where it seems to work across a variety of benchmark data sets really well. So that is this. That's what it is. We call this whole approach as MELON. You don't have to stress on the first M. MELON stands for multi-view embedding with late fusion. So here are the results. Those who attended the benchmarks know about the molecule net. But what we see is that when we compare our multi-view model, it performs better than any individual model, any individual view encoder, majority of the time. That's what the a circular polar plot is representing. And we tested this. Molecule net has 13 tasks, and there is also this cytochrome P450 set of classification tasks. And then there is also a downstream tasks dealing with GPCR, but it doesn't matter. Across a whole variety of tasks, both classification and regression, the multi-view model that we have seem to outperform the single-view models most of the time. And what we also see is a lot of robustness. There is a lot of variability, a lot less variability in model predictions when we combine the latent spaces. That's the benchmarking, but that's not the whole story. We have our collaborator here, Dr. Feishang Cheng. We are working with them closely to take this model and to test it in a real-world setting. And here, as you know, GPCRs play a critical role in Alzheimer's disease. So we tested a set of compounds. We took a set of assays dealing with compounds from drug bank and metabolites that are important for things like toxicity and metabolism. And what we are seeing is that our model's predictions correlate very highly with what you would see in a binding setting. So this is all this and more is described well in our preprint that's already out. So let's just move on to how you can see what you can see in the Hugging Face. Oh, no. So what we have open sourced in the Hugging Face is a pre-trained model and 13 fine-tuned models. You can see the details about them here. And there is also this spaces. You can navigate to that. If you click on it and go there, you should be able to test the model for all the 13 different fine-tuned models. So you can click on it. I'm just going to attempt it. Hopefully the demo gods are with me today. It takes a while because it spins up a Docker container and does all those things, but you should be able to test it. So in Hugging Face, you can get the embeddings and also test the predictions on a small scale of data. But if you navigate to, let's navigate here, our GitHub, we have given the full code for training. So you should be able to, let me see, it's very slow. So if you go through this README, it shows you how to download the checkpoints, how to do the fine-tuning with your own data, and how to verify the model performance for all the checkpoints that we have open sourced. So all our results should be completely reproducible at this point. If not, we also have put out a GitHub issue template. You can raise an issue, and someone is looking at those issues and addressing them. Thank you so much.
Okay, next we're going to get on to the hands-on lab. Has everybody managed to log on to the MOC?
The tiny URL?
Tiny URL. All right, so if you want to get started on that, I'll take you through what you're going to experience. So what we've done, my team and the Shri, who leads the development group and product group, we've got Dean and Dan and Brian around here. That can come and help you if you have any problems, all right? Let me tell you about OpenAD framework and what we're trying to achieve. So across IBM, we have a range of different models, biomedical models. There are other ones on the materials side, and a range of historical models, as well as third-party models we're looking at. And we're looking at easy ways to enable people to view them in a standardized approach, because as you move on to new architectures, the APIs are different, and not everybody's a computer scientist. So we created an approach that embeds either via an API or a command line or via magic commands in a Jupyter notebook, a framework that can easily access models, whether they're sitting on your desktop, or in this case, we're sitting up on the MOC cloud. And it gives you the ability through the toolkit to access multiple different models sitting out on your OpenShift platform, access them in this case through OpenShift AI on the MOC cloud through the workbench, which is what you'll see there from the tiny URL. And you can directly access a range of models that are catalogable within the framework you've been deployed with. And across that, so some of them could be running on your desktop, some of them could be running up on OpenShift and OpenShift AI, and you can catalog them no matter where you are in the world. As you can tell by my accent, I live in Australia. And, sorry, I'm not speaking into the microphone. And you can access these from wherever you are. You don't have to be local to them. It'll run efficiently, combined in a shared cluster, scale out as you need to within your organization. So whether you're running hands-on labs in a university, learning how to use and work with different models and run different experiments, or you're running them just yourself, you're able to log on, access the models, and go through them in a notebook via an API or via a command line interface. And we also link into a wider range set of services that can provide you molecular information. Now, the models, as you go through, you'll notice that they're defined in such a way that they're focused on computational chemistry, the model command. So get property. There are other ones that we don't have here today, whereas you can generate data sets, so for data set generation that you can call. So our goal with the OpenAD framework, which is an open source program, is to engage with more people across the community, get more people using a variety of different models, starting off with the biomedical models, and also work with us to wrap other models that they're interested in. So for instance, one of the models we've recently wrapped is a reInvent 4. Okay, which is out, as well as things such as IBM Regression Transformer and a group of other models such as MMELON we also have wrapped. And you can call them through these interfaces. And you can start and build those on your desktop using containers and scale them all the way up to, in this case, on the MOC cloud, where you will be accessing the same models that will scale based on the based on the number of users that are asking the questions of those models at any one time. So it's very efficient, it scales well with your business, and it's very easy for users to use. So to get started, you should all, through the tiny URL, be able to get onto this notebook. The first section will be just telling you about the models, where we've embedded a lot of the information that Partha and Mikhail were just talking about. And then you'll go down to the hands-on lab section. In the hands-on lab section, it's really just going to be click through. So click through each one of the tabs in the hands-on lab section. You'll go through cataloging your model, all right? And then through some demonstrations to show that your catalog is working. And then from there, you'll go through a series of exercises through visualization, running inference against both the protein and the small molecule models, okay, that we have cataloged there, and be able to go through and run a very small arbitrary triage experiment at the bottom where we filter out based on molecular weight and lithicity properties of the actual small molecules, and then go on to visualize the proteins as well that we're working with in the example. There's a lot more that you could do, but with 25 minutes, unfortunately, there's only so much we could go through today. Be very interested in your feedback now and after. I think we'll be sending out a questionnaire later on. And please put your hand up and ask any questions. We're here to answer questions for the next 25 minutes until the other demonstration comes on of the agentic workflows. And yeah, please feel free to talk to us and ask any questions as you go through. Okay, so you should have clicked through to these two screens, and once you've gone through those two, chosen MS Keycloak. If you've got the temporary ID that you've been provided because you don't have a formal MOC logon, there will be another tab below MSS Keycloak which says local login. You'll use that one. All right, but for all of you else, use the MSS Keycloak. Click through allow selected permissions and you should be straight into the notebook. And if you want to find out or participate in the OpenAD project, here are several links you can go to. We're installable via PyPI into your own notebook environment. We have our Workbench container that runs on OpenShift AI and on Podman. And we'd be really happy to talk to any of you that wish to wrap your own models using our framework, as well as interested in using or contributing to the toolkit as we move forward. We're looking to make it a real community project. This is sort of like a soft launch for us today. Yeah, so please put your hand up if you have any questions. Okay, so once you're in, I would suggest, in the nature of time, go straight down to the hands-on lab section, and you jump straight into, if I can get to that, this is too quick, the activating services. So it will get you to import the URLs to the services, which are an environment variable in your workbench. It'll try and uncatalog them just in case they were pre-cataloged, recatalog them. Then from there on in, you can just click down through the notebook. You'll see here that your services are connected. And that's an example of how you use the help for the tool, and you can look at different commands that are available. And then you'll proceed through doing a sample of inference first against the small molecule model, or MMELON, and then we'll do one against protein. One thing you'll note later on in the demonstration, we'll have an array of molecules in our molecule set. You'll be able to drill in, if I can get my, got it. You'll be able to drill in on the molecule, visualize it. We have a similar thing for proteins that you'll see right at the very end. Right, you can navigate it, and these are also available for you to click out and view as a separate set, because I've already run this before. It's updated with the full set of molecules we loaded, and you can view in, and you'll notice down here after the synonyms. So as you see, we're adding, as you page through, you'll see it add molecules to the molecule set. We can load them from SDFs or CSVs as well, or generate them separately using a generation command. And when we do it, the approach that we've taken here, we actually go out to PubChem. We pull out in the PubChem data, and what we're doing as we go through, you'll notice a number of the commands will say @merge moles, right? And what that's doing is merging in the properties into the molecule set, and then you can actually visualize the properties that we're generating as part of the process here. And you'll notice that as we go down further, this thing's very sensitive, through the demonstration. So here, when you get to the preparation stage, they're just the molecules and proteins that we've chosen. You'll notice here you can put them in as a SMILES, you can put them in as an INCHI key, or as their synonym or one of their formal names, and we'll go and look, we use the PubChem API to look those up for you. Clear our cache, and then you'll notice down here, this is where it goes and adds all those molecules on demand, singles out ones that you may have duplicated, so you've got a unique set, and then takes you through the flow. Then right at the very end, as you go through the triage step and have generated your molecules, we go to working with proteins. So what we do here is we do a DTI, drug target analysis. All right, and you can page across to the results over here. So for each SMILES, we go off and do an inference against the protein for drug target interaction. And then later on, if I go down a little bit further, you can see the protein view, and we pull this information off the PDB database, all right, directly. So you have to be connected when running this notebook to the internet. It will pull down the information, and like the other one, we can split this off into a separate tab. It'll fetch it, and you can visualize it and zoom in and rotate it as you want from there as an individual molecule. We're hoping to expand it so that we can manage sets of proteins and add properties to it like we do with small molecules. We're also looking at docking visualizations as we move forward. Okay, so I'll leave you to it now. Please put your hand up if you have any questions or any difficulties, because I know it's a very short period of time. So I want to make sure that you get the full experience, and you can go away if you've got the permanent account and play with it a little bit more when you get back to your office. Thank you. So I've been asked to explain how the small molecule inference works. Be closer to the mic. All right. I'm going to point. I should, yeah. Okay, so what you can see here, this is probably a little bit more complex style of request, but you'll see here small molecule, or SM, as the prefix name that we've catalogued the command with, or the service with. So all service commands for the SM inference will be prefixed with SM. And then we say get molecule property. Now we can identify a single property, right? These are case sensitive. Or you can do a list like we have here, and I'll show you an example of it running without a list. And here, because we've already loaded our molecules, I can say app molecules, which is a shortcut. But if you notice, if you go up just after we connected, I showed for some very simple ones as you go through the notebook, where you could just say, get molecule property BACE for ethanol, CCO, okay? And then you don't necessarily have to merge it back with molecules, but merging it back with molecules updates our molecule set. And you can see here this is the raw list, but it's also been merged back in. So when I go back down to this visualization of one of the molecules and I browse in, you can see that BACE score had already been loaded into the properties. Okay, so I'm not having to do that as a second step. But the simplest version of the small molecule inference, that's an example of the help there for that get molecule property. And it takes you through, whoever wraps the model has control over the help, so gives you a way of dynamically loading the help, but you can predefine it as the model cataloger or the model wrapper, how the help is presented and what examples you give, etc. Now, if I go back up further to the beginning, sorry, I'm just going through slowly because this is a lot faster. You can see here's the example of the get solubility for this protein, so get protein property. And up the top here for small molecules, we have get property liphophysicity for, and this molecule here, and then merging it with our molecules, all right, and then showing the actual molecule through the molecule viewer. So that's as loaded as you want. I've done it into the hundreds of molecules, but obviously it depends on your compute capacity, etc. We just introduced, so you'll notice that as you went through, sometimes things sped up. We've introduced caching as well. So one of the things that really is coming to our mind as we go through this is both performance. So particularly in your class like this, you don't want to be waiting around to recompute if it's already been computed. So you may actually doing batch after batch and some repetition. So we're looking to get our agents running in the cloud caching the results if you're asking for the same result. Obviously, you're not going to do that if it's a random seed generation that you're doing. But for things like properties, making it cached. One of the things we have in our backlog is also, which is a common question that comes up, is are you going to support asynchronous inference? And the answer to that is yes, that's in our backlog of things to do, so that you, even if you're scaling out across pods, all right, have multiple people using the system, we'll report back, hey, this job number's been submitted. Here's how to check to see whether your inference run is completed. And if you can, then you can pull the results back and then we work with it from there. So that's something in the future that we're looking at. But our whole goal is to really make it very simple so that no matter which model you wrap, you could be wrapping multiple different models. The only way you'll differentiate them is possibly by the parameters, the help, but we'll use a standard format, just like querying a database table. We're trying to keep it simple and formalized. That means you could run multiple different models or versions of the same model, all right, on your system, compare the results and pick the right results for you. Yeah, so if you wanted to run this and you're a member of the MOC, moving forward, there'll be ways for you to access the models, which is a far more efficient way. One, it's going to be more performant. Two, you're going to get the caching benefits and we'll get the scalability. All right, and you'll be either able to run it through a workbench, or if you prefer, because you don't want to put your intellectual property in the cloud, you run it on your desktop, you can just install OpenAD, run it from a notebook on your desktop. We're going to work with the MOC to put a security proxy in there, and so that you can go and get an API key. It'll enable you for a set of models, starting off with the biomedical models. You can catalog those and play around with this yourself on your desktop, but connecting directly to the MOC. Now, I live in Sydney, Australia, surprise, surprise with my accent, and I'm able to run it very well from Sydney, Australia. So I've been actually designed all these on my desktop back in Australia, and it worked fantastic, both via the workbench or on my desktop. So I'll probably have to refer to Mikhail for that one. I'm not the model person. We just take it and put it through the checkpoint.
I was talking about how nice it is here, and I didn't listen to the question. I really apologize.
The question was?
So the question is, what's the protein analysis actually doing? So there's 4 proteins. One is just a short peptide, and then the output from that is a PDB file, essentially. What am I not seeing that's happening that the system is doing with the protein inputs?
The protein inputs, it's mapped into tokens, moves into the embeddings of these tokens, and then we have cross-attention analysis with up to certain lengths. So what we learn is from the embeddings, we learn the connection between the peptide and the protein. You're talking about binding task?
I don't know.
Oh, it's from the examples we offered?
So from the examples...
Okay.
We have protein-protein interaction, right? Are you asking about that one?
So there's 4 sequences.
Yeah, that's the protein-protein interaction one.
One of those sequences is just a short sequence.
Yeah.
Oh.
Oh, yes. So to get the PDB file, we call a RESTful service out to the PDB database and pull all that information back. So we have the option that for primary information that about either a small molecule or the protein, we go off to the, I think it's there in the notebook somewhere. I'll have to look it up. You're going to have to jump up.
I don't see it anywhere obvious in the notebook. I'm just kind of curious.
All right, I'll come over and talk to you just while BC and James set up for the next demonstration.
So please continue to work on whatever you're working on, but in the meantime, we're going to move on to the last part of the hands-on session, which is a demo of how foundation models like this can be incorporated into a workflow for a real use case in drug discovery. Right? So BC is going to walk you through that demo.
All right. Thank you so much for the introduction, and I'm so excited to present the latest experiments that I want to share today. So you've heard about all these powerful foundation models that we developed. You also experienced the OpenAD demo. Now the third one that we're going to share is about the Agentic AI workflow. So here, our goal here is to really help the scientist, the third persona that Michal mentioned, to rapidly create workflows that basically leverage all the foundation models and then answer the real-world questions that we ask. So here, I'm going to use one use case. The use case here is one team of chemists who wants to discover viable and patentable molecules that are similar to ribociclib, which is used to treat certain types of breast cancer, and while reducing cardiotoxic side effects. So if you break that down, break that scientific question down, you can think of it as the team is actually trying to discover novel molecules. That means not patented before. And those molecules need to possess this CDK4, CDK6 activity. And then we should also optimize these molecules, leads, for reduced cardiotoxic effects. So thankfully, as Partha presented, we have the multi-view foundation model, which we can train it to predict the activity against CDK4/6. And we can include that as an agent to create the embeddings. So we can actually generate the similarity scores to any given input. But then it's always challenging. It requires a lot of engineering efforts to put together all these different foundation models and also connect it to the databases. And probably you also need to use other technologies to visualize them and understand the results, so which requires a lot of efforts. The simple way to interact with it, we thought, is to describe those goals in simple words, natural language, as you can see at the bottom here. So the problem here can be defined as a given molecule of the SMILE string. Find me some similar molecules that are available in a vector database, Zinc22, using CDK4 property embeddings, and check it if those molecules are ever patented. So you can imagine now, as a scientist, you can actually, like you are talking to ChatGPT, you can describe your scientific problems in words, and then the agents, generative AI, can actually figure out ways to solve this problem by integrating those available agents in databases and technologies all together and create a coherent pipeline so you can execute them and then display the results and analyze them. So agentic, AI agentic workflow, you probably heard it from the keynote today by Jeremy Jenkins from Novartis. So AI agentic workflow has the potential to realize this potential where users can just talk to this front-end generative AI, like large language models, and describe the problems in words. And then there are pools of agents that we create, and then the agents then digest these words and then figure out the appropriate agents and select them, and then also connect them to the services available or databases or other models so that we create this single sequential workflow pipeline. And then users can check each of the process and then execute the pipeline and then get the results at the end. Yes. Let me show you a live demo. So this is an experimental, so it's not actually available for you to use, but let me show you how it looks. So this is the front face. It's like a web page where you can actually see anybody's, like, for example, the studies that have been generated so far, and then you can also start from previous studies, or you can actually start from, I think I just lost my VPN, but let me see. How about, oh yeah, I come back. So, okay, so this dashboard actually shows, so we call them studies, each workflow. So we can actually go take a look at the studies that are generated by all different users. You can have your own space, and then you can go to your studies and then create a new one and name it as AI for drug discovery, and then create a new study, which then launches your experiment. Then you go to the study plan, where you can type in the prompts, just like you're talking to ChatGPT. I'm going to just copy and paste the ones that I prepared, and then enter it. What it does under the hood, what it happens is basically the agent is like a reasoning machine, digest this inputs, and then derive a detailed plan, and then divide them into well-defined tasks. And those tasks are what's shown up here. So the first task is the molecular property prediction, CDK4 prediction, and then embedding generation for similarity score calculation. And then pass that, toast that to the next one and use that similarity calculator for searching for molecules. And then at the end, we use the patent search agent to find out whether each molecule is patented before or not. So here we have the plans. Now we go to the next step, and the next step is we find all these among the pool of the agents that we created. Agents, there are so many agents, and then we select the most appropriate agents and then assign them for each task. So there we find that the fine-tuned multi-view foundation model is assigned for this task of property prediction and embedding generations. Also agent similarity search, which also uses the output of this multi-view FM to calculate the similarity scores, and then search patterns using the list of similar molecules that was generated before. So if you take a look at the flow, it actually shows the flow diagram of how the agents are assigned sequentially and how the data is being fed into each of these steps, and then provides an output, which then becomes an input for the next step. So the gray here is the data, and then blue here are all those agents that are taking the previous step's output as an input, and then finally generates the list of molecules at the end. And then finally, the next step is basically each agent then generates the Python codes that are basically going to handle each of the tasks. So here you can find the Python code to generate the embeddings and calculate the similarity scores. And you have multi-view FM codes and the similarity search codes. And then at the end, patent search. And we have two more because we need to build this workflow as a one single sequential pipeline. And also another one is to display the results at the end. All right, so you can also download this notebook as a separate IPyONB file, which you can actually take it anywhere, like you can run it in your own machine or any other computers as well. This is an experimental, so it's not available yet, but if you're interested, please come talk to us. Yeah, so, and then finally, you can execute this pipeline, which means you have all these plans generated, you assign those, you derive the tasks, and you have the agents, you have the codes available. Now the pipeline is being executed, and then it generates the results, so you can actually analyze, explore the results, and you can also check those results in the middle. The reason why we also provide all these steps made available to you is because you can also inspect those intermediate outputs. If you want to change, you can make changes. You can go back and change the prompts, or you can make changes to codes, and then you can do other things. All right, so at the end, so we get the results. You can finally see if you got the results what you're looking for. So in this result view, it actually shows a single molecule and then their embedding scores that are generated by multi-view models, and also CDK properties. And these objects basically include all other information like patents. And then if you go to the other view to display those results in a human readable format, so there's a table, and then on top, that's the ribocyclib, which is the seed molecule that we use to find the similar molecules. And if you scroll down, it's sorted by the order of similar molecules based on the CDK scores at the end. And then you can also find out the actual scores, and then how many patents are actually mentioning this. So you can see that there are actually links to those patents. So if you click on it, it actually goes and find that patent, and also scroll down to the exact page that includes the molecules. So you can check whether this molecule is indeed the same molecule. So maybe this molecule is patented, but these seem novel, which doesn't have any links to those existing patents. So maybe these two are what we are interested. Go back to the slide. Right here. All right, so to wrap up, this application, it's something that you can use and to rapidly generate the workflows. And those workflows are indeed reusable and adaptable for any use cases. And it's a way of also including the experts in the loop. So scientists actually can spend saved energy because you don't have to spend your time in engineering those workflows. You can use this kind of AI agentic workflow to generate the workflows and use the saved time and energy to really analyze and then think about scientific hypotheses and interpretation of those data that you get. And hopefully, we get to explore more opportunities to improve this kind of AI agentic workflow. And if you are interested to learn more about it, so come talk to us at the end. And with that, I'm going to switch to the next speaker. Thank you.
Hello, Leonard Lee from IBM Research. So first off, please give yourselves a round of applause for getting your hands dirty and geeking out with us. Seriously. Okay. Yeah. So our goal here is to really build, meaningful, useful tools. So, like everyone said, talk to us, give us feedback, help us make it better. Ideally they become awesome tools, right? That everybody uses. So if you miss all the QR codes, thank you. Please, the models are on the right. You're right, yes. And AI Alliance, learn more about it because we want to get people more engaged. We believe that we can build these models out in the open and really form a strong foundation. All right. Any questions for any parts of the demo? All right. Back to you.
Yeah, my question is for the demo you showed for the new small molecule which can block a drug target. So if scientists want to verify this small molecule, where they can order them or, things? Think? Okay. All right, thank you.
Zinc is a collection of molecules that can be potentially synthesized by a commercial company with a combinatorial approach. So you can just order it from enamine usually.
Okay. Follow up a quick question. So here you show the small molecule. How about the peptide, which can block a protein drug target? Can this algorithm also predict the small peptide can bind or block a drug target?
The whole point of sharing with the community is to allow much more testing. So we didn't test each and every scenario, and for peptides, we don't have yet tests. We believe the algorithm can do that after fine-tuning very well, but it's up to the community to test and share.
So we have to adjourn now because we have to get back upstairs for the rest of the forum. Thank you very much for coming. And please don't hesitate to keep using your MOC accounts.